Just want to say good afternoon. Thank you all for joining us today for this webinar that we're having with Edgewise Therapeutics. Before we hop in to the content today, we did want to share there is an upcoming Becker Education and Engagement Day. So this kind of little postcard you see on your screen. It's going to take place on Saturday, December 22nd at 4 locations. So in Florida, California, Colorado, and Pennsylvania. And it's an opportunity for members of our Becker community to come together, learn a little bit about what's going on in the space, as well as have some engagement, meet others in your community. So we hope that if you're able to join, that you can. And it's an exciting time, for those with Becker. We have a number of natural history studies underway, and we have a few clinical trials, some of which we're going to talk today with Edgewise. And we've seen this kind of grow over the past few years and have noticed, I think if you've been able to attend PPMD's annual conference, it's been nice that we've been able to have some Becker breakouts, because there is kind of a groundswell in terms of what is moving in this space. So there will be a registration link for this Becker Education and Engagement Day. Likely by the end of the week. We are recording this and we'll post this webinar on PPMD's website in the next week. We'll make sure to have the registration link and information posted there as well if you are interested in joining. So for our webinar today, we are joined by Edgewise Therapeutics. I have Abby Bronson, the Vice President of Patient Advocacy, Joanne Donovan, the Chief Medical Officer, and Alan Russell, the Chief Scientific Officer and co-founder of Edgewise Therapeutics with us. And they're going to talk about some of their ongoing clinical trials for individuals with Becker, their open label ARCH study, and some of the findings from that, as well as the currently enrolling phase 2 Grand Canyon study. If you have questions, we are going to have an open Q&A at the end of the presentation. So please put any questions you have into the chat. We'll try to get to as many of them as we can. And like I said, we are recording this webinar. So if you have friends or family who this would be valuable to, please remind them and direct them to our webpage next week when we should have this up and available. So with that, I'm going to stop sharing this and we can pull up the slides, and I'm going to hand it off to Abby to get us started for today.
Great. Joanne, do you have the slides up?
Yeah, I had to get, yeah, there we go.
Great. Good afternoon, everybody. And I want to take a little bit of time just to thank PPMD for organizing this day for Edgewise to talk to all of you. I am just going to do a little quick introduction, and then I'll pass it off to Dr. Russell. So you can go to the next slide. So this is our forward-looking statement. We're required to put this up here. It's a lot of words, but I think the most important thing to remember is that we will be talking about EDGE 5506, which is how we refer to our compound that we're studying in Becker. And it's an investigational agent, and it is not approved in any territory at this point. So you can go to the next slide. So Edgewise Therapeutics, we were founded in 2017. We're lucky to have Dr. Russell on the phone, who our Chief Scientific Officer was one of the co-founders. We were founded to develop novel therapies for the muscular dystrophies and really improve the lives of those living with these rare muscle disorders. And we have this investigational compound, as I said before, we call it EDGE 5506. It's what its name is right now. And it's a daily oral pill. And we believe that this could potentially be used alone, or it could be used in combination with other therapies, and regardless of mutation. So that's why we're studying in Becker. We're also studying it in Duchenne. You can go to the next slide. So just a little of sort of orientation in the Becker muscular dystrophy landscape. And as I think, all of you living and experiencing this disease, that there has not been much research in Becker specifically. We know that it's a serious dystrophinopathy, and what we've learned, and I think that Dr. Donovan is going to go over a little bit of this natural history data and when she speaks, but declining function, like when you start to progress on the trajectory, sort of getting weaker and less mobility, it can begin at different ages, but we have data that says that once it begins, individuals living with Becker, you do decrease every year. You're on this irreversible path to losing muscle, and then that impacts how you can function and do your everyday activities of life. And then at the same time as we entered into Becker and started studying this, we really realized that living with Becker poses multiple challenges that go beyond just the functional difficulties. And so as we have been studying this disease, it's even more important for us to listen to the community and try to understand your lived experience with this disease. We've done a lot of qualitative interviewing, which means just like one-on-one focus groups or having discussions. We have a Becker patient council that gives us input, but understanding what is most bothersome to you, what do you want to see in a treatment, what your experience is like, that has really helped us study this drug and shape our trials. And we also know that in Becker, the damage happens from contraction-induced injury in fast muscle fibers. That's a key aspect of the mechanism of the disease progression. And we have a way that we think we can protect these muscle fibers so that we can slow the functional decline. And Dr. Russell is going to talk a little bit more about that. So you can go to the next slide. So just to orient you also to the trials, as you go through this talk, you'll hear these names being referred to. So just trying to understand what they are. So I'm going to start from the bottom, actually. ARCH was the very first study that we did in Becker, and it's now on its second year. It's open label, which means that there's no placebo, no dummy drug that we're comparing to. Everyone is getting EDGE 5506. We're still collecting data, but we're not adding any more patients to that. And then the second study that we did was Canyon, and that was looking at ambulatory and adolescents in Becker, ages 12 plus. And in this one, we did have a placebo, or we did have a control group to make comparisons to. And that one, like ARCH, is active. We're still collecting data, but we're not adding in more patients to it. Then we have going up, and we have MESA, which is what we call an open label extension. And that is after you have finished a study, if you wish to continue to receive and have access to EDGE 5506, you can enter MESA and continue to receive that investigational therapy. And finally, the active and enrolling study, Grand Canyon, which Dr. Donovan is going to really go over in detail so you understand that. That is enrolling ambulatory adult males, so you have to be over 18, and it will be an 18-month study, and so we are currently looking for patients for that. So anyhow, that's just sort of an overview of the trials that we've got going. So now I'm going to hand it off to Dr. Russell.
Thank you very much, Abby. It's usually Alan, but Dr. Russell, that works. So I have a very limited number of slides today in the essence of time. I'm just going to give you a very, very quick overview of 5506 and where it stands in relationship to dystrophin and muscle contraction. And then I'll hand it over to Dr. Donovan, Joanne, to tell you about clinical data. So as Abby inferred in her introduction, thank you, some muscle fibers are more susceptible to damage due to the lack of functional dystrophin. And these fast fibers that we often talk about if you've seen this presentation before. So we're made up of 50% fast muscle, 50% slow muscle. There's a number of scientific studies showing that fast muscle fibers are more susceptible to the type of injury that you get if you have Becker muscular dystrophy. We've made an investigational therapy, EDG or EDGE 5506. And it's designed to protect those susceptible fast muscle fibers from damage, regardless of what the background mutation is, because this is a compound that specifically targets how muscle contracts, not what type of muscle, okay? In diseased animal models, and primarily what I'm talking about here is actually Duchenne animal models, but I think they are relevant to Becker muscular dystrophy as well. EDG 5506 protects those susceptible muscle fibers, and we see all sorts of good stuff coming from that, largely from the reduced breakdown of those muscle fibers. And then in clinical trials to date, Joanne will give you some of this data, we see very similar changes in terms of muscle injury protection as read out through injury biomarkers and also trends in functional improvement as well, and we'll give you an oversight to that. So Often you see dystrophin represented as the image on the left, where it's this kind of static object inserted into the membrane of muscle. But of course, muscle contracts. And on the right-hand side, I've just drawn a very simple graphic of where dystrophin is. So dystrophin exists within these green stripes. It spans the membrane, the dystrophin glycan complex. Dystrophin itself is inside the muscle membrane, inside the muscle, but the whole complex is designed to connect muscle fibers together. And you can see how it does that in two scenarios, where either all of the muscle fibers are contracting, or in this case, one of the muscle fibers is contracting, holding all these fibers together and helping them support each other when they're contracting. So if you have Becker or Duchenne for that matter, and the complex is not functioning properly, essentially the end product is that when muscle fibers contract, now they are not supported by their friends. All right. And that means that all of the stress comes along the fiber rather than along and across the fibers. And when muscle fibers get stressed, the membranes get stressed. And when your membrane gets stressed, it initiates a cascade of events that turns on the destructive pathways that prune out what they see, what the body sees as these kind of dysfunctional, sick muscle fibers. Now, that's part of the adaptive process. If we go to the gym and do weights, that's how we grow bigger and stronger muscles. But of course, if you've got Becker muscular dystrophy and the complex is not functioning properly, then the volume of disruption is too high. And that's where the inflammation and the fibrosis set in. The body cannot keep up with the level of destruction that's going on. Okay, so our approach is very, very simple. And that is to change how the muscle fibers contract in order to change how they destroy themselves. Okay, so fast fibers are preferentially injured. I haven't got data today to show you that. You just have to take my word for it. And contraction causes excess degeneration. So what we've done is design 5506 to specifically target the myosin in fast fibers, which causes the contraction. And in doing so, we've designed a selective inhibitor of that myosin to decrease contraction of the muscle fibers such that they're not injured as much. The goal is to just adjust the contraction a smidge, just to turn the volume down on the stress, if you like. And in the next slide, we'll illustrate this in action. Hopefully.
I know. This one's.
We'll get there in. Here it is. So what you're looking at here is a lumbrical muscle. That's a finger muscle from the Duchenne mouse, the MDX mouse. The left-hand one has not been treated. The right-hand one has been treated for 1 hour with a low concentration of 5506. And what that is doing is targeting the myosin in that muscle, which causes the contraction. The amount of 5506 that we've applied tweaks the contraction down in that muscle by a little bit, by 15%. And that is enough to take the stress edge off the muscle. So you can see on the left-hand side, the fully contracting muscle is getting up a full head of steam in terms of the stress. You can see dramatic fiber breakdown in these muscles. And that's these enzymes that are being activated, snipping up the fibers and essentially taking them out of the picture so that the body can grow a new muscle fiber. On the right-hand side, we've not changed dystrophin in any way. All we've done is applied the compound, is changed how the muscle contracts, not whether the muscle contracts. It's still contracting. It's just that the level of contraction now is not enough to initiate the injury response. So if this was in a whole body, the goal would be we take the medicine, we get enough compound in the medicine, it changes how the muscle contracts, it decreases that contraction injury, and long-term it should decrease breakdown, inflammation, and all the bad things that lead to the muscle replacement that causes the functional decline in Becker muscular dystrophy. I'm going to hand over now to Joanne, who will tell you all about clinical trials and data that we have to date.
Good afternoon. Delighted to speak with you. So I'm going to tell you more about our trial that is now actively enrolling, the Grand Canyon study. And I'm going to tell you some of the background information that we've gotten from the ARCH study. We have, as Abby mentioned, we've finished enrolling Canyon in adolescent and adults, and that's going to give us important information to be able to analyze and make the Grand Canyon study even stronger. So what I'm going to start out by is, how do we measure this? This is really important in a clinical trial. And how do you know that the disease is progressing in a way that we can measure? We can tell you, we can go to the regulatory authorities and share that and be able to show that a drug works. So we're using the North Star Ambulatory Assessment, the NSAA. And that includes 17 different activities that measure function in individuals that are ambulatory, can still walk. And every activity gets scored by a physical therapist, a trained evaluator, to whether or not it can be done in the usual way or with an adjustment because of weakness. For example, rising from a chair. Do you have to push with your arms on the arm of the chair, or can you get up without that? Or can you not do it at all? And that's scored 0, 1, 2. And the maximum score is 34. It's used by clinicians, and it's also used, as I said, in clinical trials to measure motor function over time. So as Abby mentioned, once the muscle is gone, then function is declined. And it's not that the muscle can't contract, as Alan just showed you. The MDX muscles, they contract just fine. But what happens is there's damage, loss of muscle, and that translates into loss of function. So as the North Star goes, as loss of function occurs, the measurement that we make with the North Star goes down. And what we see with MRI measures, a non-invasive imaging to be able to look at the leg muscles, we see that the muscle is replaced by fat and fibrosis. We know, and we'll come back to this, that in natural history studies, the North Star decreases an average of about 1.2 points each year. But as you get from not having to compensate down to 20 even, there's a lot of compensation that happens. And then you get to the point where people aren't able or off their feet. So we are able to see that even over a year. Now, what we did was we took EDG 5506 into the clinic. And as you know, we start with a phase 1 study in a limited number of people, controlled circumstances. We learn about safety. In the case of EDG 5506, we saw that it was well tolerated. We saw that it could be given as an oral tablet once a day, and it's absorbed with or without food. And importantly, we saw that it gets to the muscle. It's very concentrated in the muscle. So it's just affecting the muscle, treating the muscle, and not so much it doesn't interact with other parts of the body, basically. It gets to where it needs to be to protect the muscle. So what happens when those muscles that Alan showed you get injured? They release protein biomarkers. The membrane of the muscle gets injured, and it releases biomarkers that we can measure in the bloodstream. These include things like creatine kinase, CK, that you're probably familiar with. That's something that's measured quite routinely and often used to diagnose muscular dystrophies. Myoglobin, a particular protein called fast skeletal muscle troponin that particularly reflects the muscle fibers that are most easily injured in Becker muscular dystrophy, as well as other proteins. So we can use that in clinical trials to understand the effects of the molecule. And what we saw after 12 months of treatment with EDG 5506 in the ARCH study was that we saw creatine kinase, CK, decrease by 37%. We also saw the fast skeletal muscle troponin, which is specific to those muscle fibers, go down by just about 80%. So that was very encouraging. What was even more encouraging is we saw that these men in the trial who were at a point in their disease where they were declining, they had a relatively affected North Star of about 15, so halfway on that scale. And what we saw over a year is that instead of declining, as we would have expected in the natural history studies, they were stabilized and even trended upward. So that is important because what we hear is that it is important to be able to stabilize the disease to prevent further loss of muscle. That's what we're aiming for. So what did we learn from that study? We saw that the drug over long periods of time, well tolerated at different doses. We saw that there was rapid and sustained over 12 months, statistically significant decreases in many biomarkers of disease progression, not just the ones that I showed you. And we saw from function in the North Star and other measures like the 100-meter walk-run, the 100-meter time test, we saw stabilization of functional assessments. So we didn't see those going down. And we even saw trends towards improvement. So that gave us the information to design a potentially registrational trial. We got additional information from the Canyon study. As I said, that's going to be available in a little over a year's time. So the Grand Canyon study is a pivotal study. And what that means is that we can go to regulatory agencies with this. It's a large enough study that we can show them potentially how EDG 5506 works on function, not just biomarkers. The patients that we are enrolling, and we learned more about this from what we've done already in the clinic, are males 18 to 50, and that is because we know the most about natural history of that group of patients. They need to have a genetic diagnosis of Becker, a mutation in the dystrophin gene, and they are ambulatory, and there are other tests that would be done at screening, like the 100-meter time test, and they to fulfill those criteria as well. They are able to use a cane or other assistive devices to complete that test. And what our goal is to enroll 120 patients with Becker at about 50 sites across the United States and Europe. Now, the study, one of the things that when we learn more about safety, we can also use that to make the study more accessible, more patient-friendly. So the study has 3 parts: screening, the treatment period, which is 18 months, and follow-up. And we've been able to space out the visits, which are to look at safety, to look at function, and they are every 3 months after the first, right at the beginning, and then stretch out to every 6 months. So we've tried to minimize the visits to the site, and we also have measures that we can look at home to assess patients. So there is a randomization of 2 to 1. So 2 patients will get EDG 5506 daily tablet, and 1/3 will get placebo. As mentioned, we already have an open label study that's enrolling. It'll be enrolling the patients in ARCH, and ultimately it will be also enrolling the patients from Canyon and Grand Canyon to allow them the continuation of the drug. We'll be looking at functional assessments, which include the North Star and other things like time function tests. We'll be looking at blood tests for biomarkers. We'll be also looking at MRI, which is a very sensitive way of looking at the damage that happens over time. And so that's an important part of the study as well. That's only at the beginning and at 18 months. We're not using a muscle biopsy. There's no specific mutations that are enrolled in the study. It's open to all mutations with Becker. We've tried to have a lot of sites across the United States. And this shows the sites that are already actively recruiting. We've got at least a dozen actively recruiting. And there are additional sites that we anticipate will be enrolling later this year. We also will be opening sites in the US and Europe later, the beginning of next year. So travel is completely supported in terms of all of the usual airfare, whatever needs to be done in terms of supporting so that it's financially neutral for folks. And we've heard that from the community. We've heard that when we go to webinars, we go to weekend events, that it's important to minimize the trial burden and reduce the barriers to trials, to really support design of trials that can be done with minimal disruption of people's lives. So we have a concierge that works with folks to prepay expenses and get resources to ease the participation burden of families. And as I said, we're trying to make sure that we have more sites because to ease the travel burden for folks. So to summarize, EDG 5506 is being developed for Becker. It's also being developed in a separate studies for Duchenne. It's an oral drug intended to preserve and improve function in Becker and Duchenne, regardless of mutation. The goal is really to address the fundamental problem in the disease, that the muscle is damaged by contraction. And we are looking to protect the most susceptible muscle fibers and prevent that damage from turning into fat and fibrosis and impact the functions that you use in everyday life. We see this as being able to be used alone, ultimately also, particularly on the Duchenne side, to be used in combination with other therapeutic approaches for dystrophinopathies, and really go get at the fundamental problem, stop the damage where it begins. So we started out by telling you it's a serious dystrophinopathy. You know that already. And while function can start to decline at different ages, once it starts, it does continue. That's what the natural history tells us, that it does continue with loss of muscle and consequently function. So we've been out there listening to people to try to understand their lived experience and make things easier to participate in clinical trials that ultimately are there to help the community and get to potentially a treatment. The fundamental thing we're attacking is contraction-induced injury. It's key. And we think that protecting these muscle fibers can slow the functional decline. So we have clinical studies, both for Becker and Duchenne, that are enrolling now. And we'd be very happy to answer questions. The email is up there. I see that there's more questions in the chat function. We have also places on the web for you to be able to get in touch with us. So Eric is back on for the questions part.
Yeah, thank you, Joanne, and thank you, Abby and Alan, for a great presentation. And you covered some of the questions that were submitted ahead of time and in chat while you were going through, so that's helpful. But I think it's still good to go over a few of them. So I already mentioned that the trial is recruiting. In terms of the sites, you had that nice map of kind of the current sites that are available for families who are maybe wanting to know or track when a site closer to them is open. Is there a good way for individuals to keep track of that, of where those different sites are?
We update on clinicaltrials.gov, and that shows the sites switching from not recruiting yet to recruiting. The other is we're available to answer questions about what site it would be enrolling and most convenient for folks as well to get in touch with us.
Great, thank you. This one is a little bit less of a question, but so it was asked, but you addressed this already. Someone was asking that at the PPMD conference, you had mentioned trying to reduce burden in terms of the clinical trial design. And I think, you showed that very nicely. 7 visits over 18 months is pretty minimal for a clinical trial that we've seen in our space. And so I think that's just, appreciated given this population have families, have jobs. So taking time off to participate in a clinical trial is a challenge. And so just appreciate the thoughtfulness and design there. We also had a question come in ahead of time asking about kind of our understanding of essentially genotype phenotype correlation. So if I have, a certain type of mutation, how much do we know from natural history studies to predict maybe the changes, and how are you guys looking at that within kind of your study design?
So there are some of the natural history studies out of Italy have shown there are certain mutations that don't seem to show decline, that do maintain their North Star. We're not selecting people or excluding people based on their clinical their genotype. What we saw is in the ARCH study is that we tend to get the people who have mutations that tend to progress. So it's on a functional basis that we are enrolling folks, not on a mutation basis. That being said, there's symptoms above, even for folks that have the highest North Star. So how do we look to the broader population? And I don't see it yet, but I mean, what about the non-ambulatory folks? So first, we are looking progressively in the clinical development program from adolescents, adults. We have a clinical development program that is enrolling younger boys with Duchenne 4 and up. We'll get information, which is what we need to be able to look at safety in that population and the effects of the drug. And younger patients, 2-3-year-olds, they're on our minds too. How are we going to get to them? When are we going to get to them? So it's something that we are putting a lot of thought into. And how do we get there the fastest? Really, we see the urgent need.
Yeah. Thank you. And that was a question that we had about what is, Edgewise plans for looking at younger. So it's good to see that, something that you're considering and thinking about actively. With the site visits, there was a question for the Grand Canyon study. What is the length of time of those visits in terms of they single day, multi-day?
They're single day. That's not to say that they can't be spread out if it's more convenient that way, but they're designed to be single day.
And then with the MESA trial, is that an indefinite extension, or is that time limited in terms of that open label period?
Yeah, right now it's an 18-month study, and what we typically do is extend it as time goes on. And so rather than go to ethics committees with a 3-year study, at this point we start with 18 and then we extend it. We started ARCH with 12 months, we extended to 24. That's gradually as we accumulate more data, more safety data. That's key to that.
And then I know you showed a slide that was sharing a little bit that this original study that you did, the drug was well tolerated, within the Becker individuals. But there was a question around what are the side effects that you have seen in the studies?
Yeah, that's an important question. Sorry, I didn't touch on that. The most common side effects that we have seen have tended to be in the first few days of going on the drug, and tend to be a dizzy feeling. We give the drug at night. People sometimes get sleepy. It's about it's been about a third of the patients. It's transient, and importantly, it goes away as you continue to take the drug. So it seems to be more of something to get used to. There are structures in our muscle that feed back to the brain about where our limbs are, and we think that affecting those may be one of the reasons people have this funny feeling at the beginning. But fortunately, it does go away as the drug level stabilizes.
Thank you. And then from the submitted ahead of time as well as in the chat, we did have a few questions wondering if EDG 5506 has any impact on heart function.
I'm going to go
Yes.
to Alan here.
It's a common question because we talk about fast fibers a lot, fast fibers. But of course, individuals with Becker and Duchenne as well get cardiac problems. Now, we don't directly affect the heart, but we do believe that by changing whole body skeletal muscle health, you will have positive effects on the heart. So preclinically, we tested this in a variety of mice model of Duchenne called the DBA2J mouse. So most people do studies on the regular MDX mice. These mice are specifically designed to have more fibrosis and cardiac problems. So we did long-term studies in those mice. We did a couple of studies. And in both of those studies, we saw skeletal muscle health improve. And as a result, we also saw cardiac muscle health improve. And we saw increases or decreases in fibrosis. So that's the hope. It's a very difficult thing to demonstrate clinically because the pace of problems that occur in the heart is quite slow. But our hope is by improving skeletal muscle health, the heart will also benefit from that too.
Are there any cardiac outcomes being tracked in any of the studies, whether the Becker or the Duchenne?
We are measuring cardiac echo over time. These things, they change slowly, so that's only at every year or year and a half visit.
And then, Alan, speaking of some of the preclinical models, there was a question asking about kind of the distribution of EDG across different muscles, and if that's something that you've been able to look at. You obviously had some of the biomarker data from your other study to show that it's getting there.
Yeah, so in the preclinical models, we have the luxury of being able to take muscles out and measure how much compound is there. And as Joanne mentioned, it concentrates in the skeletal muscle. So if there's a fast fiber in that muscle, the compound will concentrate in that fast fiber, right? So in humans, in the early studies, we actually had the muscle biopsies in both healthy and Becker folks. And we saw that the compound, again, concentrated in the leg muscles that we measured. It should basically go to any muscle where there's a fast fiber, which in a human means every single muscle in your body, because there's no muscles that aren't a mix of fast and slow in the body. So it should go to every single one of your skeletal muscles.
Mm-hmm.
And then we had a question that was around, and I think you kind of already touched on this, Joanne, but in terms of kind of an approval pathway, so for the Becker population, the Grand Canyon trial is being set up as your pivotal study.
Yes. And for the Duchenne study, which I didn't talk about in this, we have other webinars that we can refer you to that speak to the Duchenne studies. The ongoing study is a 3-month placebo-controlled study with an open label part to it. And it's designed to see how the drug is absorbed in younger boys, to see how they're responding in terms of biomarkers. And with that will allow us to design the phase 3 study. What we're anticipating is initiating that study towards the end of next year. So, but as you mentioned on the approval process, there's a bunch of things that need to be done in the middle, like talking to regulatory agencies and getting their buy-in, that kind of thing. All of that administrative type stuff, but needs to be done.
And then one more asking kind of about the different doses, and is there an optimal dose that you have landed on for your pivotal study?
So what we found was that at a dose of even the 10 milligrams, we saw the maximal effect on biomarkers. That seemed to do it just well. And we didn't see any additional benefit from going to the higher doses. So with a drug, you always want to use, as you know, what you want to get this sweet spot. You want to use the right amount of drug, but not too much. And that's what we have come to with a dose of 10 milligrams for the Grand Canyon study. So there's only a single dose in the study. as I said, we're studying what the right dose is for kids and adolescents as well. So more on that, more to come.
Yeah. And we did have some questions as well, kind of around some of the Duchenne trials. I was trying to make sure we got to all of the questions with the Becker studies first. There was a question around a few around other therapies. Kind of one of, would this be something that would be a replacement to a prednisone or Emflaza, or do you see this more as kind of combination therapy and potentially something that'd be more synergistic?
We think that this is going to the fundamental problem. Right now, we are studying this on top of steroids because steroids are the standard of care. It's something that I think every clinician that works in this disease would love to see the steroids gone. So, but that's down the road a bit once we understand better how the drug works. And you also asked about other drugs. And I saw a few questions on gene therapy go by. We have been working on how to incorporate that, and I would say stay tuned on that. So more to come.
Yeah, no, I think that's definitely, it's an evolving space for us, and it's exciting and promising and great to have multiple therapies potentially for patients, and hopefully we see some expanded labels for some of these therapies that could also benefit individuals with Becker. But then it does make challenging questions for clinical trial design. So we'll want to continue to engage you on that as you all learn more as well.
It seems like it's a disease that is going to require more than one thing. And so we are looking at it in the Duchenne study. We're looking on top of exon skipping therapies if the boys are on those as well. So there's no reason to think that wouldn't already also be the case for gene therapy.
I see a question about doing weights for a 13-year-old. And this is a common question that I get and that we hear from Becker people is how much and what kind of exercise should I do, right? And especially when you're young as a Becker person, you feel somewhat invincible, right? You know you've got a problem, but it's not really manifesting, and how active should you be? I think there's quite a bit of data to say that exercise is good if you are a Becker person. It's good to be fitter by being active. But saying that, doing certain types of exercise is going to injure your muscles more than a normal person. And if you do weights in a traditional way, the whole act of doing weights is actually designed to injure your muscles in a controlled way in order to build strength and volume, right? So particularly those types of lengthening exercises underweight are injurious to Becker muscle. So I would recommend exercises like swimming, cycling, kind of less impact exercises than things like weight lifting. You'll be fine for a while, but as Joanne showed, progression is an inevitable endpoint with Becker, and you really need to protect your function as much as you can. So that would be my recommendation.
Thanks, Alan. And there are some other interesting studies going on at the moment looking at the use of exercise in Duchenne, but I think some of that as well could likely be extended to Becker as we continue to learn more and try to understand. And then there was a question of, do you think this drug would also benefit females with dystrophinopathy? That's obviously, we're starting to see more companies such as yourselves, work in the Becker space. We're getting more natural history. We have natural history studies with carriers that PPMD is funding and has helped to support. Is this something you think could also help those individuals who are having some symptoms?
So it's the same disease process, not enough dystrophin to protect the muscle. So the hardest thing in doing clinical trials is also having a good sense of how the disease progresses. So I applaud PPMD for doing those kind of natural history studies in females that are affected with dystrophinopathy. So I don't think we're quite at that stage yet, but I can definitely see in the future that this, would be something to look at in women with dystrophinopathies.
And I think that's the most of the questions that we had submitted before, as well as most that were online. If you have any other last minute questions from anyone in the audience,
there is one question about what we do when we get, how do we take care of severe side effects? You want to just comment?
Okay.
On that?
So in terms of severe side effects, the investigator is the one who is the person to call if you have questions, if you think something is going on. So you call the principal investigator, call the coordinator, they'll get you in touch. And what they do is they then consult with several of us that are involved in safety monitoring to make sure that, do they want to know if there's something else that has been seen in other patients, how we think it should be managed, all of that. So that's the kind of process when there is some kind of a severe side effect. So fortunately, the safety profile has been very good up to now.
Thank you. Question around there. We have had another one come in, Alan, in terms of expectations. So I think this is a little bit more, we could almost take it from the translating it from preclinical models, and what you were looking at to kind of, in human is almost more of the question that we can go for here. It was specifically asking if it was performing better, worse, or the same as you were expecting.
Yeah, it's a great question, that, isn't it? Because I'm in a unique spot where you kind of come up with an idea and then it goes into trials and you see what you get. Of course, we don't have placebo-controlled data yet, so I can only comment on what we have. And I might sound kind of sadly biased, but I try and be as realistic as possible when putting together a scientific story, that you keep your expectations humble so that you're always pleasantly surprised. And one thing that has occurred with 5506 is I've always been pleasantly surprised. This thing has worked better than I've anticipated all the ways along. Doesn't mean it's going to continue to do that, but so far so good. I really, the goal was to design a medicine that was applicable to everyone, that had low side effects, and so far that's where we are, but we shall see.
And then we did have another question about someone with a younger child who has more severe Becker in terms of eligibility for this trial.
Yeah. So we know less. It goes back to the natural history. We know less about younger kids with Becker. There haven't really been good natural history studies. What we can say, though, is, and we know there's this group in between Becker and Duchenne. It's kind of a continuum in terms of how much dystrophin you have. And so by developing in both, ultimately, this is something that can be used across the whole spectrum for those folks that kind of got left out of those. What we are looking at from a safety point of view is in younger boys, but I hear your question, and I don't have a great answer for it at this point. So, but it's good to bring up those questions because it makes us go back and think about it and how to bring things forward.
Yeah, and I think this goes to as well our continued need to help support natural history studies to give us the kind of information so that drug companies can develop clinical trials where high likelihood to show that drugs are safe and efficacious. But hopefully with the work that you're doing, if things go well and we get to approvals, that this all is supportive towards broad labels, which is what we always want to see in Duchenne with these therapies and Becker. And I think so with that, I just want to remind everyone as well, before we kind of sign off, that again, there is the Becker Education and Engagement Day on December 2nd. So we are going to have, again, look for those registration links. I think towards the end of this week, we should have it up when we rehost this webinar on PPMD's website, and make sure that's there for you to find. But Joanne, Alan, Abby, thank you for joining us today. That was a great presentation, and thanks for taking all of the questions. And yep, we'll keep everyone updated as new things come along from Edgewise. So thank you all.